Here are my conflicts of interest in this. They're not really related to the current topic. And my research support is as follows. The first question is, does stretch of the lung cause release of mediators? And, well, the answer is yes, it does. But you've got to be wary of short answers in complex scenarios. One of the initial studies in this whole field was from Art Slutsky's lab at Lorraine Tremblay in the JCI in 1997. This paper made a big splash. It showed that the use of, in a non-perfused, isolated lung, the use of very high tidal volumes and low PEEP, particularly if pre-treated with lipopolysaccharide, reduced in the elaboration of an extended array of pro- and anti-inflammatory cytokines. The next question is, do mediators result in injury? Well, this was the basis of a theory written by Dr. Slutsky and Tremblay, which suggested that not only are they released from the lung, but these are not bystanders, these are pathogenic. The idea of biotrauma was initiated, and the concept that not just the lungs but also distal organs would be involved. Therefore, the ventilator becomes a motor of multi-organ failure. Well, in patients, this was actually demonstrated. Marco Ranieri and colleagues in a small study looked at control versus protective ventilation. And you can see here the BAL on the left. Just get the little pointer out there. Pointer doesn't work, never mind. Where my mouse is here, you can see the BAL on the left and plasma on the right. And broadly speaking, you see a mean effect of not much change or a lowering in cytokines in the presence of protective ventilation, but in control subjects, an increase. But what you notice with this is that whilst the averages are positive, there's a broad range of values. There's also a broad range of responses, really saying that these patients are extremely heterogeneous. So there's a signal there, but there's also a lot of noise. Well, not everyone agrees with the idea that mediators are pathogenic in ARDS, and the group of Dreyfus and Simon, the first author in this was Jean-Damien Hucard, and they published a series of papers that fundamentally disagreed with the whole idea. The first slide here demonstrates in vivo and ex vivo that 40 mils versus 7 mils per kilo really does cause injury, so that's a sort of a control experiment or demonstration. And the second here in ex vivo without LPS pretreatment demonstrates that while there are low levels of interleukin-1 beta and MIP2, there's no TNF detected, and there's not really much difference between the low stretch and high stretch. So this was a kind of a controversy that went on backwards and forwards in the pages of the Blue Journal. In the meantime, there was an important paper by by Dr. Imai, looking at a combination translational paper looking at the effects on rabbits in vivo and also human data. Here you can see peroxidase tunnel, that's a tunnel stain demonstrating apoptosis, that is prominent in the injurious ventilation, but not in the non-injurious protective ventilation. Here you can see the apoptotic index is high in the kidney and in the villi of the small intestine. These are organs that have high cell turnover and high metabolic requirement, and so are very vulnerable. And it's important here that in those rabbits, they're very susceptible to injurious ventilation. But the lung, paradoxically, in fact, showed that the opposite was the case. So that was a sort of an inconsistency in that overall idea that the mechanical ventilation would injure distal organs, but perhaps protect the lung against apoptosis. And in the human data on this very influential study, what you can see here is a correlation on the y-axis of changes in plasma creatinine from baseline against change in plasma soluble Fos ligand. Fos ligand is one of the main steps in the pathway leading to classic apoptosis, and you can see the open symbols show lung protective ventilation. Really, the creatinine went down, and in the non-protective ventilation, the creatinine went up in rough correlation to an index of apoptosis. So if you believed cause and effect here, you would say that as far as the kidney is concerned, the kidney is being damaged by apoptosis, resulting in decreased renal function, and that is caused by the high tidal volume ventilation. But cause and effect are pretty tricky concepts in biology, and they're very tricky concepts in applied clinical research. Koch was the discoverer of the tuberculosis, I was going to say vaccine, tuberculosis bacillus, and Robert Koch, German, and he elaborated the idea of Koch's postulates, where you have cause and effect and trying to establish them. And Austin Bradford Hill, who invented the randomized controlled trial, made major contributions in this, looking at more modern ways of trying to nail down cause and effect. And nowadays, we have blockers of all sorts of processes. We can knock down RNA. We can use genetic knockout mice. So we're kind of closer to nailing down cause and effect, but it's always very, very tricky. We asked the question, could circulating mediators cause villi? We thought there was an open question, and Thomas Jacklin, one of our research fellows, applied sort of Koch's hypothesis to this in a discussion paper. The first postulate is microbes must be found in disease but not in health, and he sort of said, well, circulating factors must be increased by injurious ventilation and not by non-injurious ventilation. That will be one. We'll prove it and support it. The microbes must be isolatable from a diseased organ and grown in culture. Well, soluble candidate mediators can be derived from the lung and identified, so they're there. The next, cultured microbes can cause disease when introduced into healthy organisms. So if you collect the perfusate from an adversely ventilated lung, could you cause injury in a gently ventilated lung? And then the same microbe isolated in the inoculated disease experimental host, the correlate here is an increased candidate mediators found in injury caused by transfer of the perfusate. So this is a picture of Thomas Jacklin, a Swiss fellow who worked with us, very earnest and always impeccably punctual. Swiss, so very good, not like the Irish fellows. And this is his kind of scheme here. And you can imagine a perfused mouse lung. So you take a mouse and you cannulate the pulmonary artery and you drain the left atrium. If you don't collect the perfusate, what you can see if particles or mediators go into the perfusate, it simply disappears into the drain. Here is a schematic of the recirculating perfusate. You actually collect it and reinfuse it. This is what happens in the body to an extent. And what you can imagine there is that if anything, you don't have to know what it is, if anything is elaborated into the perfusate, well, its concentration will increase with every circulation. And if you don't believe me, it's drawn on the schematic. That was a joke, by the way, but fair enough, you're a tough crowd. So I'm going to show you a series of slides here where the ventilation was either high or low, high being bad and low being good, and the perfusate was either recirculated or not recirculated. So here you can see on the y-axis is an index of lung injury. Now, there are many indices, and they're all very consistent. This is the one I chose to show in the next several slides. It's protein leakage. Higher protein leakage, more protein seen in the BAL. On the y-axis, there are two conditions, the tidal volume, which is either low or none, here, just with fresh perfusate, and the presence or absence of recirculation. So what you can see here is that with low tidal volume, it makes no difference whether you recirculate or not. It causes no increase in the BAL protein, no injury. If you use high tidal volume and don't recirculate the perfusate, then you get substantial injury. If you get high tidal volume and you recirculate the perfusate, you get more injury, quite consistently more. And so we reasoned this amount was due to stretch, and could not possibly be related to circulating anything because of nothing being recirculated. And this is due to whatever is in the perfusate. We didn't know what that was, but that's what it's due to. So the next set of experiments, because Thomas was Swiss. He didn't take any chances. He wanted to nail this down perfectly in order. He said, what we will do is we'll ventilate the animals on one day with either high or low tidal volume. We'll collect the perfusate and we'll store it. And then the next day or the next week, we'll ventilate with protective ventilation, low tidal volume, but we'll use the collected perfusate. And so here you can see on the y-axis is lung injury, again, and on the x-axis is the ventilation. They're all low tidal volume recipients. So these two got either perfusate from a low tidal volume donor or fresh perfusate, and there's no injury. But if you ventilate with a low tidal volume and you get perfusate from yesterday's high tidal volume, you get a lot of injury. That can only be due to something in the perfusate called mediators. Don't know what they are. We used an ex vivo prep so that we could rapidly get a signal as to whether the epithelial cells were intact or not. The transepithelial resistance, kind of like an Ussing chamber type of setup. It gives you a quick readout, so you can expose the cells to the perfusate or to any kind of solution, and you can see what it does to the resistance. If it lowers the resistance, that means it's impaired, and that correlates well with dysfunctional leaky capillaries. So here you can see when we exposed the high tidal volume perfusate, there was a substantial reduction impairment. So high value means big impairment compared with fresh perfusate. Of course, fresh perfusate spread over the epithelium impairs its resistance. But when we diluted the perfusate, we saw that the more dilute was, the less impact this had. So there was definitely a dose-response phenomenon going on there. We next looked at particle size, to see were these small molecules or big molecules causing the effect. Heat lability, most proteins that are complex are highly heat labile, and then whether there was any extractable lipid using a Falk lipid extraction. So we found here, you can see here fresh perfusate and high tidal volume perfusate. When we used a 30 kilodalton filter, we found that the ultrafiltrate, the bit that was filtered, and the retentate each had a comparable contribution to the inhibition of the transepithelial resistance. The second thing was heat lability. If we heated the perfusate, it abolished the effect totally. And then the last thing was with lipid solubility. Here's fresh perfusate, and this is no added fraction, but if you extract the added fraction, if you extract the lipids and then re-add that extracted lipid to fresh perfusate, what you see is that there is a proportion of this that is lipid soluble. So what we have is a heterogeneous potentially lipoproteins, potentially, or a mixed bag of compounds. Now, we spent an enormous amount of time trying to find out what was the magic factor, because we thought we could sell it and hurt everybody's lungs, or we thought we could sell it and find out how to remove it. We then said, okay, well, what about the usual suspects? What about, mediators that everybody talks about? Maybe we can't identify it from first principles, but maybe we can just measure the concentrations of known mediators, and it would explain things. So here's TNF, and you can see here on the y-axis is the level of TNF, and on the x-axis is high versus low tidal volume recirculated versus not. So what you get are very low levels. Now, you've got to be careful comparing levels with different preparations, but they're low levels of TNF, and the only thing that makes the TNF high is collecting it over a longer period, the recirculation. Second, if you draw a graph against the injury on the x-axis, BAL protein leakage, and the level of TNF in the perfusate, there's no correlation whatsoever. So in this setup, where injury was certainly transferred, it was not transferred by TNF. So present, but not responsible. Doesn't mean it's not responsible for injury and pro-inflammatory effects elsewhere. Now, lipid mediators are sort of seeing a new dawn in the whole area of lung injury. And you can see this graph here, and I'll show you 3 graphs of important lipid mediators that are known to be pathogenic under many circumstances. This first one is thromboxane B2, important for increasing permeability and platelet aggregation. And you can see here that with high tidal volume, with recirculation, and without blocking cyclooxygenase, there's a lot of thromboxane B2. High tidal volume, recirculation, and use of ibuprofen substantially reduced, so it is made in this system, and when you block its synthesis, it's made a lot less. And then low tidal volume with recirculation and no ibuprofen is a control level. Pretty well the same pattern, varying degrees, but the same pattern present, abolished, for PGD2, an important precursor, and then prostaglandin E2, exactly the same pattern. So this looked fairly promising, and then we looked at the functional impact of using ibuprofen in this circulation, and it made no difference whatsoever. So we have a lot of things that we can see and demonstrate, but we can't put our finger on what it is that causes the damage. So the question then is, where is the injury occurring? It's another way to look at the idea, could circulating mediators cause damage? Well, here's non-dependent aerated lung and dependent atelectatic lung, and we've demonstrated in rat models, Shin and Tushita demonstrated this, that with injurious ventilation, taking regional biopsies in paralyzed ventilated animals, all of the injury, the bulk of the injury occurs where the ventilation goes in the non-dependent lung. And this was reproduced very, very well, I thought as well. The same findings were found using PET scans in patients who were paralyzed by Giacomo Bellani and colleagues here in Milan several years later. And the reason that's relevant to this talk is that if you think that the perfused cytokines are responsible for injury, well, you need more than perfused cytokines, but certainly it does not occur where the perfusion is greatest. That's not a very strong argument, but it's an argument. Then, is this all new? You're supposed to come to the SMART meeting to find new information. Well, you saw new information in the last presentation, but this idea of cytokines and mediators is not remotely new. John Edmonds, middle author here, was the first author of the first description of this work in 1969. And what did they do? Well, they published this as a full paper a couple of years later. They wondered, because they found that patients during surgery, or patients who had shock, some patients in ICUs, because ICUs were being developed then on mechanical ventilators, in fact, were hypotensive. And they didn't quite know why that was. It could have been the positive pressure causing reduced preload. That was beginning to be understood back then. But also they thought there might be humoral mediators. And what you can see here is a strip of lung on the top of the screen and a strip of intestine on the bottom of the screen. And every time the lung is stretched, the intestine, which is sitting below it, has a contraction. And using methods demonstrated by Jeff Vane and colleagues, they're able to suggest that this droplets, in fact, contained prostaglandins, and that was potentially a clinically relevant mediator, mediating, they didn't call it multi-organ failure, but hypotension and organ dysfunction in patients who are mechanically ventilated. So this isn't new at all. What's my opinion as to whether this happens? Circulating mediators are not a sole cause of clinical ventilator-induced lung injury. I sort of believe that. There's no basis yet to antagonize or to remove mediators, but I wish there was, because they would make a big difference. And I think there's realistic hope for the future, not just based on this work, but based on gene profiles, for example, expression profiles and proteomics for patients with ARDS. Thank you very much indeed.